Hello students. Students, today we are going to discuss a very important topic, and that is genetic code. So let's see what is genetic code is. Genetic code is actually the relationship between the sequence of nucleotides present on messenger RNA and the amino acid in polypeptide. In my previous video, we saw that the genetic information is copied from one strand of DNA into messenger RNA. And this process is known as transcription. And the information which is present in messenger RNA will lead to the formation of polypeptide, which is known as translation. So there is relationship between the sequence of nucleotides present on messenger RNA and the amino acids in polypeptide. My dear students, the term genetic code, it was given by George Gamow. Now George Gamow suggested that the genetic code is triplet in nature. Genetic code is triplet. It means the codon contains 3 letters. For example, I am giving you an example of a codon, that is GUG. GUG is one codon and it codes for the amino acid valine. Similarly, we can take another codon, that is UUU. It codes for phenylalanine. Clear? So he said that it is triplet in nature. Genetic code is triplet in nature. We see that there are 4 types of nucleotides, and these 4 types of nucleotides has to make 20 types of amino acids. So they are triplet in nature. It means that how many codons are there? There are 64 codons. Clear? So the term genetic code was given by George Gamow, and he said that these codons or these genetic codes are triplet in nature. Now next important scientist is Marshall Nierenberg. Marshall Nierenberg is famous for his cell-free protein synthesis. What he did, he made a messenger RNA. And this messenger RNA contained only one type of nucleic acid, that is uridylic acid. Now here we can see, my dear students, that this messenger RNA contains a sequence of nucleotides which have only one type of nucleotide, that is uridylic acid. Second important thing, as George Gamow suggested, that the codons are triplet in nature. So this is our first codon, this is our second codon, and this is our third codon, and so on. So he prepared a messenger RNA having only U, that is uridylic acid, and after the translation he saw that the polypeptide which was synthesized, it contains only one type of amino acid. Today we know that the codon UUU, it codes for phenylalanine. So here we can see UUU, it codes for amino acid phenylalanine. Similarly, UUU, next codon, it also codes for phenylalanine, and the third codon, it also codes for phenylalanine. So he saw that the codon UUU, it codes for this amino acid, and this is the first genetic codon was cracked. The first codon was cracked and it was codon UU and U. The second scientist is Dr. Hargobind Khurana. Now what Dr. Hargobind Khurana did, he also prepared a messenger RNA. He synthesized a messenger RNA Now he took two types of nucleotides, that is U, uridylic acid, and G, guanylyl acid. Uridylic acid and guanylyl acid, and so on. And after translation, what happened? A polypeptide was synthesized. This polypeptide contains two types of amino acid. UGU is the first codon and GUG is the second codon. The third codon is again UGU and the fourth codon is GUG. Now here, as we know today, UGU, this codon codes for cysteine, the amino acid cysteine, and GUG, this codon codes for valine. So here we got a polypeptide having two types of amino acid, UGU, that is cysteine, and GUG, that is valine. UGU cysteine and GUG, that is valine. Clear? So what Dr. Hargobind Khurana did, he made a synthetic messenger RNA, and when he was making messenger RNA, he took only two type of nucleotides, that is uridylic acid and guanylyl acid, and he got two type of amino acids in his polypeptide chain. The third scientist is Severo Okawa. Now, what he did, he made a synthetic mRNA without any template. He did not use any template. And for this, he used an enzyme, and the name of that enzyme is polynucleotide phosphorylase. So these scientists contributed for the genetic code. In the next part, we will see what are the salient features of the genetic code is. Students, now we will see the characteristics of genetic code. The very first characteristic is that genetic code is triplet in nature. Clear? We saw that there are 64 codons. Why there are 64 codons? Because there are 4 nucleotides and they are present in triplet, and we saw that it leads to the formation of 64 codons. Out of these 64 codons, only 61 codons code for amino acid and 3 codons do not code for any amino acid. Those 3 codons which do not code for any amino acid, they are known as stop codon. These are also known as nonsense codon. So the very first characteristic is the genetic code is triplet in nature. Second is universality. The genetic code is nearly universal in all the organism, whether it is prokaryote or whether it is human being. The same type of codon will code for the same type of amino acids. Third is it is unambiguous and specific. Unambiguous means that one codon will code for only one type of amino acid. But there is some exception. We will focus on exception. The codon GUG. It codes for 2 amino acids. I told you that codon is, or genetic code is unambiguous and specific. It means this codon GUG, it should code for 1 amino acid, but the exception is that GUG, it codes for 2 amino acid, that is valine and methionine. So you have to remember the exception case also. Third is, we have studied unambiguous. Now fourth, it is commaless. Commaless means when the genetic code we will study, we will see that these codons are present in a continuous manner. For example, UGA, UU, UAGG. Now there we cannot see any space between them, neither there is any comma. They are written in a continuous manner. So they are commaless. The genetic code is commaless. There is no punctuation present. We have to see first three letters. They will lead to the formation of the first codon. Now next three letters, this will be the second codon and this will be the third codon. Next is the rule of degeneracy or degeneracy of genetic code. Degeneracy means that one amino acid can be coded by more than one codon. For example, here we can see the amino acid glycine. It is coded by the codon CUU, CUC, CUA, CUG, UUA and UUG. So it means it is degenerate. The genetic code is degenerate. Serine, it is also amino acid and it is coded by the codon UCU, UCC, UCA, UCG, AGU and AGC. So this tells us that the genetic code is degenerate. Degenerate means one amino acid can be coded by more than one codons. Here we can see there are how many codons? 1, 2, 3, 4, 5 and 6. 6 codons can code for leucine. Similarly, here also we can see the amino acid serine. It can be coded by UCU, UCC, UCA, UCG, AGU and AGC. How many codons? There are also 6 codons. So these 6 codons can code for one amino acid. But there is also an exception. We can see the codon that is UGG, it will code for tryptophan, and AUG, it will be coded by methionine. So these are two amino acids, tryptophan and methionine, which is coded by only one codon. Clear? Next is the chain initiation and chain termination codon. When there is formation of polypeptide, there is the codon which start the formation of polypeptide, and that is AUG. So AUG, it performs the dual function. First, it is required to start the synthesis of polypeptide chain. So that's why it is known as start codon. Clear? The codon AUG, it is required to start the polypeptide synthesis, so it is known as start codon. Beside this, it also related to the synthesis of amino acid, that is methionine. So it has dual function. It codes for the amino acid methionine plus it acts as a start codon. Clear? Next is there are 3 amino acids. As I told you earlier, there are total 64 codons, and out of these 64 codons, only 61 will code for amino acid. 3 codons will not code for any amino acid, and those 3 are UAA, UAG and UGA. My dear student, you have to remember these 3 codons, and these 3 codons, whenever they come in messenger RNA, the synthesis of polypeptide will be terminated, it will be stopped. So that's why it is known as stop codon. It is also called as nonsense because they do not code for any amino acid. So here, these are the characteristics of genetic code. I think now this topic is clear to you. Thank you.